So I think probably it's a good time to get started. I want to welcome everybody. Hopefully you can all hear me. Welcome you to the LSO Networking and Webinar Series, Knowledge Webinar Series that we've been doing. A couple of housekeeping issues before I introduce our speaker. So if you have any questions, I think the most appropriate way is we'll probably do questions at the end unless something pops up that's of vital interest right then that Catalina sees. And so if you could put them in the chat mode, I will moderate those questions as we get to the end. So, but put them in as you are thinking of them so you don't forget about your questions as we go along. What I wanted to do today is introduce Catalina Lopez-Correa. So Catalina, as well as being a colleague and a friend, Catalina is one of the foremost people in genomics in Canada and actually globally. So she's currently the executive director of the Canadian COVID Genomics Network, which is led out of Genome Canada, CanCOGeN. It's a huge multi-million-dollar federal initiative that Genome Canada is responsible for, which we'll probably talk a little bit about today, Pan-Canadian, which is looking at sequencing the viral and human genomes of COVID, try and help inform public health and policy decisions so that they're science-based, based on what's going on with the information of the virus at the time. We're lucky today to have Catalina actually coming in to us from Colombia, where she was previously the Chief Operating Officer at Ruta in Medellin. And in this capacity, she led the operations of a world-class innovation hub that really looking at technologies and approaches to solve the city's most pressing both economic but also social problems. And I would say that that's something that Catalina's been passionate about throughout her career. She's also championed several initiatives prior to this, looking at the impact of genomics in the developing world, including a book on genomics in emerging economies and a white paper with the World Economic Forum and several articles. And since early 2000, she's worked as an evaluator for the European Commission. I first met Catalina when she was Chief Scientific Officer at Genome Quebec, part of the genomics family, as we call them, the Genomics Enterprise, which is a set of genomics centers across the country, including Genome Canada. She then moved to Genome BC before going to Colombia, and we are very privileged, actually, that she has decided to come back to Canada. She has a medical degree, a PhD. You can read the whole thing in the bio that was provided, as well as many awards for her outstanding participation in the field of science. So I'm going to pass it over to Catalina, and we can all listen, and I'll be back to moderate the Q&A at the end. So I'll pass it to you, Catalina.
Well, Alison, thank you so much for this nice introduction. Yeah, so it's been a long journey in the world of genomics with Alison and so many other great colleagues working in this field in Canada. So it is a pleasure for me today to present the work we are doing with CanCOGeN. So what I would like to do this morning is to walk you a little bit through the work we're doing with CanCOGeN, and in general also how genomics can help us understand and control the COVID pandemic, and how this is tied, is linked also to the work we have been doing in Canada around precision health and precision medicine. So we can start here, what I think we have all really known already for several months, that we are all basically on the same storm. We're all facing a pandemic internationally, globally. Is really a global challenge, but we are not exactly all on the same boat. Basically, we see some people that will, get the virus and be asymptomatic, some other people that will be severely affected, so not everybody is affected in the same way. We also see many challenges from a social and economic perspective that we're not going to discuss much today. But yeah, all this to say that we're all in this facing this big challenge, but we're not all being affected in the same way. And what is interesting, what we're going to discuss today, is how science and technology can help us understand this difference and can help us control potentially the pandemic and understand why some people maybe are affected differently and how is the behavior of the virus. So among those many different technologies that we have available, there is one that has been a key area where Canada has been investing for years and years and years, and that's the area of genomics. So in genomics here, we see that we often talk about the informatics revolution, and I like here to talk to you about the genomics revolution. And if we compare the blue line that we have in the screen, which is representing the Moore's law, Moore basically predicted that computers will double their power, memory and processing power every year, and the cost of the computer and that memory will be half every year. So if we imagine what's happening now with computers, what's happening with the informatics revolution, and we compare that with the green line, which is basically the genomics revolution, meaning how much more sequencing we can do now, how much faster and cheaper are the technologies now to read our DNA, this genetic code, ACGT, the 4 letters that make the genetic code, and that are part of every living organism. So now our capacity to read those and our capacity to understand the meaning of those, of that genetic code is really at the basis of the genomic revolution. And Canada has been investing through Genome Canada and the Genome Centers for about 20 years. Now we're actually celebrating our 20th anniversary this year. So 20 years investing in this technology, in genomics, in applying, in understanding, in getting the capacity, in getting the equipment, in getting the knowledge, getting the expertise to understand these genomics. And that's really at the basis of the CanCOGeN initiative. And this initiative basically is here getting about a total of $40 million in federal funding. 20 million of that 40 is going to be directed to viral sequencing, and we're going to go into the details on that. And $20 million will go into human or the host sequencing. And what are the goals? So the goals for CanCOGeN are basically to coordinate, to get those sequence and coordinate the efforts of the viral and the host genome sequencing initiatives. And we have an overarching goal of sequencing 150,000 viral genomes and 10,000 host genomes. So that's a significant effort. And we will see the scale, of the size of the genomes, because if we think that a viral genome is 30,000 letters and a human genome is 3 billion letters, we can just, make the proportion here and calculate the cost for both. So that's why we can do many, many more viral genomes. We want also to be able to integrate the genomic data, because genomics is, 4 letters, great, is amazing and wonderful to get to sequence the genome of all those virus and hosts. But the most important also data is to be able to link with clinical data, epidemiological data, and what we call the metadata. So that's what will give the value and help us really advance the understanding of the behavior and help us potentially control the pandemic. Now, another important aspect of CanCOGeN is to facilitate and promote data sharing. That's one of the challenges we have, not just in Canada, but internationally, in terms of how we are sharing the data. Most of the work we're doing, as I say, in Canada and international, is being in little silos. We sequence the genomes in a center, but we're not often being able to share because of either legal, ethical, regulatory challenges. The other aspect we want to work with CanCOGeN is to build capacity. So to build the capacity to prepare Canada, Canada has been, one of the countries that is reacting well and very well to the pandemic, but we need to really be prepared for the future. We need to be able to be much faster in the way we react and much more proactive. So building that capacity is a critical part of the CanCOGeN initiative. Another aspect that's, we are saying at CanCOGeN is the goal is not to get the 10,000 genomes, human genomes, and 150,000 viral genomes. That's not the end point. I mean, that's one of the goals, yes, but it's not the end point. So the end point is really to be able to inform policy and public health with that data that we are generating. And we are hoping also to be able to use this information to help advance drug and vaccine development programs in Canada and potentially internationally. So what is the structure of CanCOGeN? So CanCOGeN is a Genome Canada initiative. What we have initially is a steering committee. I'll show you, it's a very strong international steering committee with people that have been working in the COVID genomics area in the last months, from the UK, from the US, from Europe, from Canada. And we have the two initiatives, the host genome sequencing project with an implementation committee. You can see who are the members and the chair for that initiative. And we have the viral genome sequencing project or initiative with an implementation committee, and also the members across representing different provinces and different expertise, being clinical, being epidemiological, data, genomics, all that, bioinformatics. So, and that's basically the structure, but the power of this initiative is that it's not just a standalone initiative. It's really working in very close collaboration and hand by hand with the provincial health laboratories, with hospitals, research institutes, large-scale genome sequencing centers, industry coordinating centers, and of course, you know, when I say Genome Canada, we're talking about Genome Canada and the genomic enterprise. So all the genome centers across the provinces are also an integral part of this initiative. Now, this is the steering committee. As you see, you know, we have representation from many different institutions and people that are working on the UK initiatives and others. So it is a very strong steering committee that is advising us, is helping us guide this initiative. Now, what is the real need that we have now? So I think one of the key needs is to be able to coordinate the host and the viral genome sequencing efforts and coordinate that with the data collection. So I think we have with CanCOGeN a unique opportunity to really link clinical data, epidemiological data, host and viral, and to make this right and just get the basis for the future work we could do, and potentially for new outbreaks, if there are, and for potentially new pandemics. Now, CanCOGeN also gives us a unique opportunity to influence policy and public health decisions. So to just use this data, and I'll show you some examples on how the data, genomic data, is not just, you know, cool data that can be in papers. Of course, there might be papers out of this work. There might be nice articles and lots of data. But the end point is not just an article or a nice paper in a high-impact journal, but it's really to be able to influence policy, to be able to influence public health decisions. And it's also an opportunity for us to help us understand from a human genetic perspective how our genome is making us more susceptible to have different expressions of the disease. And all these tailored to Canadians. So understand in the Canadian population how our genetics and our genomics is affecting the way we respond and we actually are affected by the virus. The other need is to be able to work on a global level. You see, the virus doesn't see borders. And yet we are keeping our data, of course, because of security, because of privacy. We keep it in silos, in countries, in regions. So we need to find ways, while, you know, preserving security and privacy, being able to share this data at a global scale. And I'll show you some examples on the work we're already doing in that data sharing at a global scale. And again, I go back to building capacity, which is a very important aspect of the work we're doing. Now, let's start with the virus. So the virus, let me see, it didn't, okay, here it goes. So the virus, sorry for that. So the virus initiative is, we're gonna go here a little bit in details on what we're doing and what is the key aspect of this viral initiative. And I will highly recommend this New York Times April 30th article. It's open access. And it's written by one of the most amazing scientific journalists, Carl Zimmer, and also Jonathan Curran. And they have an incredibly interactive and graphic, presentation where you can see the mutations, how they interact, how they move around the world. So it's a cool article. I highly recommend it. It's an easy read. I would say it's really almost entertaining. I mean, it's not a scientific article. It's really a New York Times for everybody to read. So I highly recommend that. And that really what is explaining us is how this virus is mutating and what the variants of the virus are and what do they mean and how can we analyze. So what we know is that, as we all know, the pandemic started in China in December, and we can see in orange the L variant. That was the first identified variant. And after that, we see this basically phylogenetic tree where there are many other variants that are being developed. We have now around 7 variants of this virus. There are some that are super close related, the G brand, the GH, GR, G, and G just by itself. So we are understanding that just because of the sequencing we're able to do. So now we're able to sequence the entire 30,000 letters of the genome, and we can see how this virus is behaving, when it's mutating, how is it changing, and all that, which is, again, thanks to genomics that now we can do all this analysis. So this analysis is helping us understand how the virus is moving around the world and what, on this movement, what are the variants that are more prevalent in certain regions and how those variants are interacting, how are they spreading, how they're evolving. So all this is basically, now we can do it just thanks to the genomics work we're doing. So with this, we can see now what is the frequency of certain variants. We see that in Asia, the L, the S variants are more frequent, whereas in North America and South America and some parts of Europe, more the G variants, the blue, green variants are the most frequent ones. So again, all this is helping us give a global picture and a global understanding of the behavior and the movements of the virus. So all this data is being deposited in a database called GSED, and it can be visualized by another tool that's called NextTrain. So today there's more than 80,000 viral genomes, whole genomes that have been submitted to GSED. And that's really helping us build all these global maps. So, there's about 1,500 sequences from Canada have been already submitted, and there's about 4,000 or 5,000 that are in the process of being having the quality control and all the review to make sure the quality is excellent to be submitted. So there's many other viral sequences in the pipeline from Canada to be submitted. Even from Colombia, where I am now, there's about 80 sequences that have been submitted. So all countries in the world, being developing countries, being countries that have maybe more means to do this type of research, are submitting this data, and this is really a tool that is helping us have this global vision of what's happening with the virus around the world. So that's really thanks to the new technology, genomics, that we're now able to gather data almost in real time, that we can generate this data very, very rapidly. But there is, of course, also a risk that the accuracy and reliability and the quality of the data is not that good. And that's why in Canada, rather than going very, very fast and submitting thousands and thousands, we are, with CanCOGeN, developing and coordinating a quality control process to ensure that the data we're submitting is of the highest quality. And of course, this data is helping, and I'll show you an example right now, is helping us just make policy, very concrete policy decisions. We'll see an example in a couple of slides. So why then we need to sequence? I think you already have an idea that this is important, but we really, with this information, it's helping us understand the transmission trends of the virus. How is the virus moving at a regional, provincial, national, and international level? How we can also perform cluster investigations and see where the clusters of the virus are and get all the epidemiological data. I think many people haven't heard much about epidemiology years ago, and now suddenly it's becoming one of these disciplines that everybody's talking about. And part of that power of epidemiology can be, reached with the data we're gathering with the genomics of the virus. Now, it is also helping us understand how the virus is evolving over time, and will help us refine our detection methods, either PCR, serology, all the testing we're doing for the virus is helping us also understand clinical outcomes. In some, potentially, we have talked in some cases that maybe some strains could give some higher or stronger phenotypes or more stronger clinical outcomes or more severe outcomes or higher transmission. And it's also helping us inform public health and policy decisions. And again, I'm going to show you an example. And it's also could help us develop treatments that are potentially targeted to a certain variants of the virus. Now, in terms of policy decisions, I like to talk about this example in BC. And many of you have heard about Dr. Bonnie Henry. Dr. Henry has been leading the public health initiatives and groups in BC, in British Columbia, for the control of the pandemic in that province. And she has been very proactive and has been doing a stellar job. But what is interesting here related to the work we're doing in genomics is that Dr. Henry started to get access to viral genome data very early in the pandemic. Actually, Genome BC was one of the first centers that provided funding for viral genome sequence in BC. So Dr. Henry was using these data, and they, with her team, they started to see soon in the pandemic that there are many viruses. Of course, they had the airports closed, and they were controlling, and understanding where the virus was coming from to the province. And they started to see many, and you see them in blue, light blue, many of the variants of the virus were coming from the US, from Washington state. And they decided, based on the genomic data, and of course, based on all things too, but one of the data points that helped make the decision for closing the border with the US, and that's actually one of the slides from Dr. Bonnie Henry, when she presented how they made the decision to close the border, was based on this genomic data. So it's, as I say, genomics is not just to publish papers, which is good and is excellent, but it's also to make important policy decisions, like closing a border in this case. Now, on the host side, the host gene initiative is led by CGen in coordination with Genome Canada. And that, as I say, the main objective is to sequence 10,000 human genomes. Now, why do we need to sequence, or what is the challenge we have? And I was saying in one of the first slides that we are all on the same storm, but we're not all on the same boat. And that's one of the explanations for that, not being all on the same boat. One of the reasons might be in our genome. And here we can see how basically everybody could be potentially affected or infected by the virus, but not everybody is affected in the same way. We see that some people in red are severely affected, with some people in the right-hand little graph, to the right-hand side of some people that might have maybe more comorbidities. We hear, well, if you have diabetes, cardiovascular disease, you will be affected more severely. And those are the people in red, and the people in dark blue are the people potentially that have less comorbidities, healthier, younger, might not be as affected. But we're seeing younger people, like this little red man there to the left, or woman, that are young, potentially that have no clinical history, that have no comorbidities, and they're still developing a very severe presentation of the disease and end up potentially in intensive care units. So what's happening with maybe some people that are outliers, basically, that are either young and developing severe diseases, or maybe older with comorbidities and being exposed and not being affected. So what's happening are some people maybe that are resistant to the infection, some others are asymptomatic, some others have severe diseases, and how can we use genomics to understand why some people react in different ways. So the whole initiative, as I say, really is to sequence, but why do we need to sequence? And it's basically to understand this inter-individual clinical variability. We want to also understand the role genomics is playing in this clinical variability. There might be other factors, but genomics might be one of those factors. That also could help us develop biomarkers that could help us predict why people are at higher risk of developing maybe more severe disease, and could help us also develop more precise therapies. And basically what we're saying here with all these is that this is leading us to what we have been talking about in Canada for years, which is precision health, using genomics to understand why we're sick in a certain way, how we respond to medications, what is the right medication for us, and when and how should we use that medication. And the study will have, as we say at the beginning, several subgroups and sub-projects. Ethics is a big part because we need to get the right informed consent to be able to analyze the genomes and to get that data integrated and aggregated for data and sample collection, for sequencing. So there are several groups that are working on that as part of the host genomics initiative. And we're also, of course, working with several clinical centers. This is open to everybody, so you're highly encouraged to come and reach out to Genome Canada, reach out to me personally, if you have any studies that you think could be part of this host sequencing initiative. We have been already in contact with about 80 studies across Canada, but then we need, of course, to get 10,000 and to get the right groupings of the clinical groups, we need to get more, we need to have access to several potential studies. Now, this is what we're doing in Canada, but Canada, as I say, is not working in isolation. We have many international initiatives that are with whom we are collaborating. One of those is ICOG, is the COVID Genomics UK Consortium. This is an initiative, of course, in the UK. They're sequencing viral genomes. They're a little bit ahead of us. They have been sequencing already for a while, viral genomes. They have submitted 25,000 genomes to GZ. They got funding of £20 million for this initiative. And similar to us, they are working in partnership between the public health agencies, the NHS, the Wellcome Sanger Institute. So it's really a partnership initiative. The other initiative that is interesting is the COVID Host Genetics Initiative. This COVID Host Genetic Initiative is, again, an international initiative where there are more than 1,000 members, 194 studies, 46 countries represented. So every country can submit. It's not about whole genome. It's not about, and that's why, they could just be one gene. They can analyze one gene, one panel. So it's all about genetics, but not about genomics, let's say, not about whole genome studies. And they have two options for the scientists that want to participate in that study. One is that you could share your individual data, and that's the preferred way to do it. And the other one is that they can aggregate the data, summarize, and present the statistics. So those two ways are completely available for scientists or groups that would like to participate in that initiative. Now, the other one is the UK Biobank. And actually, we do have also in Canada, maybe not structured in the same way. We have Canada Future, we have cores like CanCOGeN, Generations and Generations BC, and several biobanks that have been built across the country that could be used in the same way as the UK Biobank to do further studies on COVID. So what the UK Biobank is 500,000 volunteer participants that are being followed, that can be recontacted, and now they are being followed to fill some questionnaires about COVID-19. And because they have sequencing data for many of those, they will be able to correlate symptoms or this questionnaire on COVID with some of the genomic data. So this is just starting, but it hopefully will give us also a lot of important data. Now, one country that you might have heard that is doing also quite an interesting work in genomics and genetics and COVID is Iceland. So Iceland is actually started investing in genomics years ago. I used to work for DecoGenetics in beginning of 2000, where they were still doing microsatellites, and soon they moved to do sequencing and basically sequence the entire population. And now they're able to use part of that data to understand some of the effect that the genome has in how people are responding or how severe the disease is in certain individuals. But what is interesting too is that they were one of the countries that started to do testing in asymptomatic individuals very early in the pandemic. And they started to test basically everybody that wanted to be tested, even though you were not sick so basically they ended up testing almost the entire population, and they end up one of these first papers in the New England Journal of Medicine describe the fact that there were many people that were asymptomatic. At the time, we didn't know exactly that were that many asymptomatic people that were infected and that were able to, spread the virus. So that was one of those first studies when they had this strategy to do the testing and to analyze the virus. So they were understanding, as you see in the little graph with the green and with the orange, the purple. So the bars, they were able to understand with the sequence of the virus, they were able to understand if that was a virus that was coming from outside of Iceland, or that was transmitted through family members, or how was it transmitted, and all that with genetics and genomics, and with the capacity of doing massive testing across the entire country. Now, all these, as I say, is leading us to think that this is not just about genomics. This is about seeing medicine and health in a much more comprehensive way. And this is exactly what Leroy Hood was talking about in the year 2013 when he started talking about P for medicine. Well, I'll explain a little bit what it is. And basically, it's a coalition. It's really a convergence of a digital revolution, and that's what we're seeing now. We are ability to use apps to track and trace where, who is infected, who is in contact with whom. Also consumer-driven and social networks. How are we building the social networks to help understand help us educate people on how to protect ourselves and all that. And systems biology, understanding the genome, understanding not just the viral genome, but the human genome, those interactions and all this. So that makes part of this P for medicine. And how will now this P for medicine, that's basically a medicine that is predictive, preventative, personalized, and participatory, how is this applied now to COVID? Here, there are some examples. So in predictive, now we see with our understanding of the virus, how we see how the virus moves around the world, how maybe there are potential areas that are at risk for an outbreak because the viruses we saw with closing the border. We understand that maybe there were viruses coming from the US, and a way to control the pandemic in DC was to close that border with the US and help control the entrance of cases. So that's a predictive. We're predicting that those maybe cases are coming from there, and we can prevent. Preventative, how we can do, social distance, individual protection, all those strategies. It's not just, again, about genomics only, but it's about public health. It's about seeing health as a continuum. Personalized is everything we're doing now with genetics, and I'm trying to understand what are the genetic factors maybe that explain the inter-individual variability in clinical outcomes. And participatory, well, this CanCOGeN in particular is a multidisciplinary, multisectorial initiative involving government, involving academics, private sector. So it's really everybody basically is participating in this initiative. Now, just to end, there's this paper that is public access, and I highly recommend it. The World Economic Forum has this precision medicine statement that was published in May this year, just 2 months ago. And it's interesting to see that some of the governance gap they basically say there that genomics is already, we're already able to read the genome. It's low cost. We're already able to, we have all the sequencers, we have the capacity we have the technology, all that is ready. Now what we have is more gaps on the governance. That is really what is a bit slowing down our capacity to fully adopt this precision medicine or precision health strategies. So some of the challenges they're seeing there is data sharing and data interoperability. And this, as you saw, is one of our key goals with CanCOGeN. We think that CanCOGeN is giving the opportunity for Canada to really think pan-Canadian and to get that data in the right way so that we can share, and that data is that we have this interoperability that we need to make the maximum, get the maximum out of that data. Now, there's also all the ethical aspects, ethical use of the technology. So here there are a few points. One, well, of course, as I say, we're working with several teams, Sparta, Nopper's team at McGill and many others, on the legal, ethical aspects of the informed consent to make sure that we do this right from the beginning, that we spend the time in having the right informed consent and that everything is right from the beginning. But there is also the ethical aspect in terms of access. We need to make sure that these technologies are used by everybody, that we include First Nations, that we include regions that maybe have more limited resources. And that's why this is not just about Canada, it's a global paper, it's global access. So that's another of the challenges we have with COVID, is that, countries like Canada, we can invest $40 million in sequencing host and viral genomes. But the virus is not just in Canada, the virus is global. And the pandemic, this one and potentially future pandemics, will be global. And that's why we need to have, a global vision on how we are addressing these challenges. Now, another very important aspect is social trust and social engagement. This is not just about science. This is about all of us. We're all affected by this virus. We're all in this pandemic. So we have all to be able to understand how the technology is helping us and trust that technology to be able to help advance those. As I say, access and fair pricing, when we're thinking about global, we have to think about all regions, all populations, all groups that could have access to that. And another aspect is a responsive regulatory system. And I think Canada has been very proactive in having some kind of regulatory sandboxes where you don't have to go through the regular regulatory approval, which is very lengthy, a very long process, but you could have maybe a speedy process for some of the challenges, some of the technologies that could help us face some of the challenges we have with the pandemic. So we have seen that in Canada, and that's part of the initiatives we're having here. And I think with that, I would just like to say thank you to all of you. I see some questions. I haven't had the time to read them, but I'm happy to answer any questions you might have on the presentation.
Yeah, that was really great. So we do have a few questions, and I'll just walk through them. Somebody had asked about sending the link to the New York Times article, which we already did. So it's already done there. For those of you who want, it's in the chat. But has there been any information already with the sequencing that's done that this variant mutation rate in the virus might affect the efficacy of the vaccines? If they're making them to certain variants, that they might be more regional than global in terms of effectiveness. Is there any information from the sequencing that's shown that yet, or is it too early?
Yeah, I think it's early. From what we have seen, none of the mutations are really affecting in a major way the function of this virus. So we don't have variants that are maybe more aggressive or more that are changing the behavior or the capacity to infect of this virus. And that leads us to think that, well, with the information we have so far, we have only 7 variants, and let's say 80,000 genomes sequenced from the virus. So it's a bit early, but the hope is that, if there are any variants, with the amount of data we're able now to collect at an international level, we should be able to see when there are variants that potentially will touch some of the key areas of the virus where, where either the vaccine or the medications that we're developing, the drug development programs that we are now working on could be targeting. So for now, I think it's early days still, but that's the hope that we could inform that vaccine development with these. Hopefully, we will not have too many variants so that every region will have to develop a vaccine, because just developing one vaccine is a monumental task. So yeah, let's hope that we can use it for most of the variants. Yeah.
Great, thank you. And so a couple of questions following on about the host genome. So Jennifer sends an important question about the heterogeneity in testing policies and how it might have impacted the data that has already been generated. So for example, in March and April in Ontario, testing was real, and I think this is true in other jurisdictions. While it's been opened up now, early on, it was really only for high-risk groups or frontline workers, or people who were tested, who were obviously positive, right? So really, and so has that affected the data? So I think there was a question before about that, about the proportionality of asymptomatic people that might be in the cohort, which would affect the kind of host genome interaction if asymptomatic people are not getting sick. So can you talk a little bit about whether heterogeneity of data across provinces is going to affect the ability to draw conclusions?
Yeah, so it's a really good question. And well, think we have 10,000, we have, funding for 10,000 samples. And it seems is a lot, but in reality, it's not a lot. When we have to divide that across the country, make sure that we cover all the provinces, make sure that we cover different groupings, clinical groupings. Certainly, the asymptomatics is a difficult group to gather because, maybe early on they were not detected early on in the pandemic. As I say, the only country that started super early to look at those asymptomatics was Iceland. Well, not the only, but one of the countries that started to look at that early. Canada didn't, and some, there were some tests potentially on asymptomatic, but it was not systematically done. Now, the other aspect that is important here is that we still, even if we focus on the symptomatic ones, we still have groups of high interest. Like, think about the individuals that are younger than 50, that are no comorbidities, and that end up in intensive care unit. That's an extreme phenotype. And we could have access to those phenotypes because those were individuals were affected, and that could show us and teach us how your genome maybe is leading you to that. We're also working with children cohorts. Potentially, we have heard about the Kawasaki Syndrome in some children. So is there any genomic link to the Kawasaki Syndrome? When they initially started Kawasaki before COVID, before we even think about COVID, there was already some genetic variants associated with Kawasaki. So it will be very interesting to see some of those genetic variants hold with that. So and the other aspect to answer that question is that it is important both for the virus and for the host that our strategy is not just to look backwards at what happened before and take the samples of patients that were diagnosed, or samples that were taken at the beginning of the pandemic, because we want to be able, as much as we can, to take decisions almost real time with the virus. So for the virus in particular, we also want to keep part of the viral genomes, our capacity to sequence on future outbreaks, so that we are able to use that data in a very quick and rapid way to help inform policy and public health decisions. And for the host, we are hoping also to get, if we don't get enough asymptomatics on the course we have previously, we have the opportunity to look in the future and now start looking at some of the asymptomatics, maybe individuals that have been closely related with and have been in extreme contact with the virus and yet not being sick. So testing some of these people and including them in the cohort.
Maybe I could just ask a follow-on question before I get to Andrew's question about, are the 10,000 virus genomes matched to 10,000 host genomes matched to the 10,000 viral genomes within the bigger cohort, so that you can look at variant and host at the same time?
Well, this is the $1 million question.
Oh, okay.
And it seems trivial. It seems the most logical thing to do. And I can tell you it's not trivial. And I can tell you we're not the only ones struggling. We had a call with the UK, and I actually had a call with Sue Hill, which is the Chief Scientific Officer for the NHS, National Health System in the UK. And they're all struggling with that because, the way samples are collected, the informed consent we get for the host is gathered in a way, is through the hospital, whereas the viral sample is going through the public health lab. Seems like a very simple thing. In my mind, when I started, it was like, oh, this is the most simple thing. Actually, this is the ideal. We're pushing for that. We're hoping to get a good proportion of the 10,000 linked with the virus, but it's challenging. I can tell you it's challenging.
Okay. And so Andrew had an interesting question again about the cohort, the 10,000, about what initiatives have you put in place that ensure recruitment and proportional representation from the typically marginalized populations within Canada, especially since some of them demonstrate disproportional disease severity?
Yeah, so we are, as I say, we're working with the different genome centers across to make sure that we're not just focusing on Ottawa or Ontario, but we're covering the entire Canada. We're working. We have been in discussions with a project that's called the Silent Genomes Project, that's working with First Nations, and they have been setting up a governance framework for working with genomes and genomics with the First Nations populations. So we are hoping to include a group of First Nations into the CanCOGeN cohort, and we're very vigilant and proactive to make sure that we include as much diversity as we can, not just in terms of First Nations, but gender. We're looking at that, and that's an important aspect. That's part of the design of the host-seek initiative. Yeah.
I mean, is there a Canadian opportunity here because we're so multicultural? So I think of in Ontario here, just north of Toronto, we have a very large South Asian community. South Asians have been disproportionately affected, and so ethnically we're quite diverse. So is there quite a Canadian, almost, I don't want to say an advantage, but an opportunity here to look at a multicultural distribution around the severity of the disease?
Absolutely, Alison, absolutely. And that's one of the greatest assets. You don't need to go far in Canada. All cities, big cities, and everywhere in Canada, we have such a great diversity that, yes, that will be for sure a great differentiator for this initiative.
Okay. From Saeed Masroor, he has a question about their startup company working on advanced machine learning solutions for genomics data, and wanted to know whether there are opportunities or grants, collaborations within CanCOGeN for companies like them.
Yes, call me. This is the short answer. The long answer is, yeah, you can feel free to reach out and contact me, seriously. I have been talking already to a few companies, and we have already some ideas on I think, as I say, the data we're generating, the objective is not to have the data on a cloud. The objective is to use this data in the most meaningful and productive way. And we need all hands, all brains, and all participation to be able to use this data in the most meaningful way. It will be great to be able to work with the private sector on that. So feel free to reach out, and we'll be happy to chat. I'm already chatting and discussing with a couple of companies on AI, on data mining, and potential biotech companies that are developing new drugs and that would like to get access to some form of the data. And the good news is that I think very proactively, CanCOGeN, when they started having, even before I joined, when they started having the discussion about the informed consent and all that, they were already thinking about the industry potential access to the data. So there is a framework where, there's potential for industry access, with certain parameters and all that. But yes, all to say is, yes, we are very interested in discussing with the private sector, getting your ideas on how you could use the data, how you could help mine the data, make sense of the data, and use it for your initiatives, or prepare the data to help other groups or other companies use that data in their initiatives.
So there you go, everybody. On the industry side, there's a call to action. So get a hold. So, you know, feel free to email Catalina and arrange a chat so you can figure out how you can participate. There's a question around, do we have any evidence yet, again, might be early, about, because most of Canada has a low infection rate, that outbreaks that we are seeing, so we're seeing a little outbreak in BC right now, and we've had other little outbreaks happening, that's driven by one variant versus another?
Yeah, well, we are starting to analyze that, but it's still early. Still, as I say, you know, we understand how the virus is moving, and when we have typically now outbreaks, most of them are from one variant. We saw that the blue-green variants, the G ones, are the ones that are prevalent in North America. But it's early days. I mean, there are some articles that are starting to appear, some studies that are starting to appear, where we start to get an understanding, but we need more data, and we need to be able to also, as I was saying, be not in the reactive mode, but more in the proactive mode. So now with the outbreak in BC, that will be a good opportunity now that the whole sequencing is in place to see what's happening with that outbreak and where is this virus coming from. They're all coming from one. Can we identify exactly where this person came from with that virus and it spread it? So is exactly what we want to do, and we're getting the capacity built across the country and the expertise to be able to react now fast and understand and use the data to understand that.
Great, Catalina. So maybe here's a good last question before I'm going to hand it back to Jason Field, who's our executive president of LSO. But what is the plan for timely communication about CanCOGeN from what's going on in CanCOGeN to keep the public informed? So obviously you've got your policy people, but particularly as the situation evolves, how can we all keep informed about what's going on with CanCOGeN?
Yeah, so first, well, you should follow us on Twitter here, use the CanCOGeN hashtag. So we will be posting on Twitter all, you know, when there are talks, when there are information. We are finalizing the setup of our webpage for CanCOGeN that was part of Genome Canada, so that we will have also updated almost weekly, or if it's even more often than that, how many genomes we have sequenced, how many genomes we have deposited. All that information is going to be in the web page. And what we're also trying to gather is more success stories, like the one I was telling you in BC. So if any of you is involved with these genomics in either the viral side or the host, and has any success stories to share with us, we'll love to have those stories to illustrate, so that people can understand that this is not just about the data. This data is actionable, and what are the actions we can take with this data? So all this will be in the webpage and our social media.
Great, thank you very much, Catalina. I really do want to thank you. It's a bit earlier over there in Colombia, I believe. So thank you very much for getting up a bit earlier. But I think it's fascinating. Obviously, I'm in the genomics field, so I find it fascinating, but really very useful about how it's actually translating into making a difference, not just producing data. So I think that's fabulous. Thanks for giving us that case study. And we'll make sure that we know when your website goes live, and Twitter, and make sure those go into our newsletter that goes out across Ontario to our members, so people know how to get in contact. So thank you very much. Thank you to everybody who submitted questions and was online. And I just want to pass it over to Jason for a couple of words at the end here.
Hi, everyone, and thanks, Allison. And thank you, Catalina, for that wonderful presentation. You know, I was really struck. I always say, you know, Canadian science is second to none. and to see the work that you're doing and contributing on a global level to the fight against COVID-19. It really reinforces all that we know about the quality and the importance of Canadian science on the global stage. So again, thank you for sharing with us the work that you're doing and the continued efforts that you're making to this fight against COVID. I have a few quick announcements. Saw a lot of familiar faces, or names, I should say, on the list of attendees. And I have to say, I really miss seeing all of you in person, but we're doing our best to try to connect with you virtually through a number of events. And I just wanted to share with you a few upcoming items that we have. Of course, there's always our mentorship platform. Again, a great opportunity to network and connect virtually. So please check that out. The links are all available on our website. We also have a fun event that's coming up. We, of course, had to cancel this year's golf tournament. So we have a virtual ungolf tournament. There's actually no golf involved whatsoever, but if you're looking for an opportunity to attend educational sessions like the ones that we just heard from Catalina, as well as to have some virtual networking opportunities, this is a great event. We also have some fun giveaways and prizes that are part of this. So please check it out. It's a fun little event, and again, a way to connect with your colleagues from around the life sciences sector, and to also have an opportunity to listen to some really engaging seminars and webinars as part of this program. In terms of our upcoming webinar series, our next one is actually happening next week on August 19th. This one is about unlocking life sciences career opportunities in these uncertain times. And we have folks from BioTalent Canada talking about some new programs that they've just launched, as well as some people, some young professionals, some experienced professionals talking about their career journeys and providing advice to young professionals that are launching their careers in the life sciences during these very, very interesting times. And finally, I just want to say thank you to our corporate sponsors, Alexion, Sanofi, Shift Health, Silicon Valley Bank, Innovative Medicines Canada, and Merck. Our gold sponsors, Taiho Pharma, Admere, Antibody, Stryker, Horizon, and BioTalent Canada. And our silver sponsors, EY, University of Toronto, Scarborough, Roche, BDO, and CRB. Again, without the support of our sponsors, we wouldn't be able to have these sorts of programs, and especially during these times where we can't have in-person events, their support is even more meaningful and important to us. And finally, I just want to say thank you to all of you for attending and participating. Again, I miss seeing all of you in person, but hopefully we will get to do that again soon. And in the meantime, encourage you to sign up for our online events. So once again, thank you, Catalina. Thank you, everyone, for joining us. Have a great rest of your day. Take care. Stay safe.